participating in this process. This is the top 10 papers reading list voting or polling from this year. Just a few notes. We set the dates for the papers to be between September 2014 and August 2015. The first stage, as usual, was a nomination stage in which about 35 papers were nominated. All of them very nice papers and very nice work. And in the second stage, the final voting, we had people vote on those nominated papers. We received 51 unique responders and with a total of 204 votes, which we'd like those numbers to be better, but at the same time, this it's not too bad. It's about 20-25% of the attendance of this conference, and so a few disclaimers as usual. Again, although it's a fantastic group of papers. In both stages of this process, we acknowledge that it's a highly incomplete list and it's not a scientifically conducted poll, and there's way too many biases possible. Then we do little to control for these biases. We just let the community have its say and have its voice. Your input is very welcome for future iterations, including people who want to run the poll in the future years. I'd be very happy to hand over the baton. So without any further caveats, and those are the top 11 papers due to a tie from the poll. I'll read them so that if people at the back can know. Quantitative modeling of transcription factor binding specificity. Please hold your applause until the end. Of transcription factor binding specificities using DNA shape. Zhao et al. From the Rose Lab, PNS 2015. Deconvolving the recognition of DNA shape from sequence. Abe et al. From the Man Lab, Cell 2015. A method to predict the impact of regulatory variants from DNA sequence. Lee et al. Michael Beer Lab, Nature Genetics 2015. Predicting the sequence specificities of DNA and RNA binding proteins by deep learning. Ali Panahi et al., Brendan Frey's lab, Nature Biotech. Inferring interaction type in gene regulatory networks using co-expression data, Khosravi et al., Bader Lab, Algorithms for Molecular Biology. Computational analysis of cell-to-cell heterogeneity in single-cell RNA sequencing data reveals hidden subpopulations of cells. Buettner et al. From the Stegler Lab, Nature Biotechnology. Joint annotation of chromatin state and chromatin conformation reveals relationships among domain types and identities, and identifies domains of cell type specific expression. Liebrecht et al. From Bill Noble's lab, Genome Research. Elucidating compound mechanism of action by network perturbation analysis. Wu et al. From the Califano lab, Cell 2015. Large-scale imputation of epigenomic data sets for systematic annotation of diverse human tissues, Ernst and Kellis, Nature Biotech. The human splicing code reveals new insights into the genetic determinants of disease, Xiong et al., Brendan Frey. Integration of genomic data enables selective discovery of breast cancer drivers, Sanchez Garcia et al., Dana Pierce Lab, Cell. A big congratulations to all the authors of these, and many of whom are present in the audience today. Thanks for your participation and your work.